leads the Systems Biochemistry group at the Digital Metabolic Twin Center. His research develops predictive biomolecular network models to study metabolic and neurodegenerative diseases. He earned his PhD in systems biology from the National University of Ireland in Galway. And it's a pleasure to have you here, Ron, and looking forward for your presentation. Thank you.
Thank you for the introduction. Do you hear me okay?
Yes, quite well. Mm-hmm.
Okay, just share my screen. Now, you see that okay without any blocking?
Yeah, it's perfect. Mm-hmm.
Okay. So yeah, so good afternoon or good morning, everybody, depending where you are. So today I would like to talk about a problem I've been working on for quite some time in collaboration with others also, and essentially it's addressing this, what I think is a paradox in genome scale modeling, where existing genome scale modeling methods, they only explicitly model reaction rate, but not molecular species abundance. So there are some genome scale modeling methods or approaching genome scale modeling methods that include kinetics, but those are, those techniques, my perspective is they're not so scalable. And so the problem is with that is that you have a situation where the experimental data measurements of molecular abundance are the wrong variables, because in your models, if you have reaction rates as your variables, the measurement variables are of molecular abundance, and there's a mismatch caused by that. So you have these genome scale models where we're modeling essentially the variable that's not been measured experimentally. Even in the case of fluxomics, what's being measured is abundance of different stable isotopes, and you have to use the model to infer the fluxes. So it's still relevant in that case. So the problem then came around is the question is really, how do you constrain genome scale models with reaction kinetics? And so there's a variety of different approaches out there. All of them, as far as I've seen, are an approximation on the actual kinetics that go on. So you can think of ln-log kinetics, or you can think of the Gecko modeling framework. They're all an approximation to the actual kinetic rate laws. So the problem with the kinetic rate laws is, of course, that they're nonlinear, and you have these high dimensional systems where you have nonlinear equations you want to include, and that presents some problems. So yeah, you can look at it in two ways. How do you constrain genome scale models with reaction kinetics, or how do you extend reaction kinetics to truly genome scale? So just as a bit of introduction here, there's going to be a lot of exponential and logarithms in this talk. So just to remind people of these identities here, the main one is, let's say, down towards the bottom here, where you have a product of these x's to the power of a. Well, that can just be represented equivalently as exponential of a transpose times the log of x. So all the exponentials and logarithms in this talk are meant component-wise. So when you use those identities, you can generate a matrix vector formulation of elementary reaction kinetics. So here in the simple reaction, water's splitting into hydrogen and oxygen. The stoichiometric coefficients to that reaction are represented in the matrix N, and the stoichiometric coefficients for the forward direction would be represented by F, capital F, and the stoichiometric coefficients in the reverse direction would be represented by capital R. So the rate of the forward elementary reaction, VF, is the product of the kinetic parameter K in the forward direction, KF, times the product of the concentrations to the power of the stoichiometric coefficients. So anyone in systems biology will be very familiar with these types of expressions, but you can reformulate them in terms of matrix vector notation down in the last two lines here, where you have the exponential and logarithmic terms. for forward and reverse reaction rate. Then the dynamic equation for evolution of molecular species concentration is given by this expression here, and the fourth line down you have fully matrix vector expression of that, which to me is interesting because it highlights that the forward and reverse stoichiometric coefficients appear both in the coefficients of the expression and in the exponents via the term inside the exponential. So when you replace the logarithmic terms there with their equivalent variables on replace the linear terms with the equivalent variables on logarithmic scale, p and x here, you get an expression for the change in logarithmic concentration with respect to time. That's the negative the diagonal of the exponential of the negative concentration, or like negative the logarithmic concentration times this f of x. And f of x is let's say would be the key function one would be trying to solve if you're trying to compute a steady state. So this is as simplified as I could find to make this expression. The difference between the forward and reverse stoichiometric coefficients, and the exponential term is then gives you your rates, where p and x are in logarithmic terms. So p is logarithmic kinetic parameter and x is logarithmic concentration. So for quite a while, I spent time studying this function and trying to characterize what type of function f of x is, because most of the mathematical optimization literature out there, they start off with a hypothesis about assuming f of x is some particular type of function. Well, to be able to plug your biological problem into those established algorithms, what you have to do is try and characterize what f of x is. So that begs the question, what type of function is f of x? There's so much literature out there on monotone functions and solving monotone variation equalities, etc. But unfortunately, f of x is not monotone. I think almost in general it is. And why I have to put that caveat there is we were able to show, so a monotone function here is the expression y transpose the gradient of f of x times the y. So that's always greater than or equal to 0 for any y, right? So if you do numerical experiments with a variety of genome scale models, you'll find that this expression does not hold numerically. So theoretically, we were able to prove for certain small biochemical networks that it's indeed definitely not monotone. And we're able to show that it's a new class of generalized monotone function, which we called duplomonotone. And so a duplomonotone function is a restriction on the set of monotone functions. So only a subset of monotone functions every monotone function will be duplomonotone, but the duplomonotone function is not monotone. So what's the restriction? So the restriction here is if you replace y with f of x, then this inequality is true. So only the y's that equal f of x will give you this expression. And so if you look for some sort of gradient type method, then to solve for f of x equals 0, you can't use the theory out there on monotone functions, at least in the ambient dimension. And so this was an interesting result at the time, but we can't prove that every biochemical network results in a duplomonotone function, although I highly suspect that it's true. So I'd be super interested if someone could prove it in general. We developed an algorithm also at the time for computing zeros of duplomonotone functions, and it works pretty well, but it didn't really lend itself to let's say integration with constraint-based modeling, and I can illustrate a little bit more why later. So fast forward a number of years, and what I have to, let's say, I found a new technique to approach this problem. And so the main thing here is that the convexity of the set you're optimizing over is what's important, and not whether that set is polyhedral convex. So most of the genome scale modeling methods at the moment deal with polyhedral convex sets, but optimization over certain convex sets, is, even if they're nonlinear, is possible. So, for example, consider this function f of x. It's convex, and the epigraph of that function illustrated in green, restricted between x1 and x2 bounds. That function is, or that set, that epigraph is itself convex. So every convex function is associated with a convex set. And for certain convex sets, then are cones, and so this is illustrated here. The interior of this traffic cone type shape structure is called a Euclidean cone, and that's the one most often used in conic optimization. So in this conic optimization approach, you're optimizing over convex sets, but they may be nonlinear, and the algorithms are still competitive with linear optimization type and the same dimension, because they use interior point algorithms to solve the problems. So there's a race on in the optimization community, it seems to me, to characterize the set of cones with convex cones, which one can easily optimize over with, or quickly optimize over using interior point algorithms. So conic optimization, as I say, is just optimization over some nonlinear but convex set. And it's actually linear optimization in general over a conic constraints. So a particularly interesting cone for kinetic modeling is what's called the exponential cone. So the exponential cone is an asymmetric cone, unlike the others, and it's defined by this three-dimensional set here. So x1, x2, x3 are the variables, and it's the cone is defined by this expression x1 greater than or equal to x2 times exponential of x3 divided by x2. So if you look at that's a very expressive cone. So you can model many different functions with that cone. But in particular, if you consider a two-dimensional slice of that cone, and you say that x2 to be fix x2 to be equal to 1, then the exponential cone just becomes the epigraph of the exponential function. So the exponential cone is everything that's above this blue line here. So x1 greater than or equal to exponential of x3. And why that's particularly interesting for kinetics is that if we recall again these matrix vector representation of elementary kinetics, the only sort of nonlinear term that we had left after logarithmizing things was this exponential term. And so what the core of variational kinetics, this new method I've been developing, is to relax the constraint representing elementary reaction kinetics, and instead of having, let's say, v forward to be exactly equal to the exponential of f transpose logarithmic concentration time plus logarithmic kinetic parameter, you relax that constraint. So then you're dealing with a relaxation of the problem, but you're optimizing over the epigraph. So that set is convex. So you can use these, as say, interior point algorithms to optimize over that epigraph, and the intersection of any number of convex sets is itself convex. So one of the convex sets can be the familiar steady state mass balance constraint here, the n times Vf minus Vr plus your exchanges equal to 0. So the intersection of the, let's say, kinetically relaxed exponential cones, which are these terms here in the bottom, and the polyhedral convex set, that's convex. The intersection between those two is convex, and so you can set up a linear optimization to optimize over it. And so the sort of naive approach I had at the beginning was just minimize along the flux direction and maximize along the concentration direction to try and hit this boundary here. So once you hit the boundary, these inequalities become equalities. So then you get a solution which is steady state mass balance, and it satisfies kinetic constraints. So this this approach, then you can really go wild with it, like because exponential cones are very expressive. You can use them to maximize entropy, for example. You can use them to get equivalence between the logarithmic variables and the linear variables, so logarithmic concentration and linear concentration. You can use the quadratic or Euclidean cones I mentioned earlier for penalization from given mean plus or minus variance experimental data, whether that's for fluxes or kinetic parameters or concentrations or standard chemical potentials. And as you all know, when you have a linear constraints, they can just be added together. So we've heard already about the mass balance constraints. You can add on constraints to make sure that the kinetic parameters are thermodynamically feasible and that you have moiety conservation. So the general schema thing there is that you create an intersection from these sets, which is itself convex. That's represented by this small set blue in the middle, and then you're optimizing towards the boundary of it, where you try to hit the boundary where the kinetic constraints or the correspondence between the logarithmic and linear constraints concentrations is exact. So it's relaxed initially, but you're aiming for the exact representation, or a numerically exact representation, which is not a relaxation up to numerical tolerance. So in the end, we're aiming for a solution somewhere on the boundary. So this approach can also be applied to more complicated functions here. For example, this convenience kinetics expression. So the limitation with what I told you before was that if we were to apply that technique to genome scale models, well, the genome scale models have overall reactions, typically, not the elementary reactions. So you'd have to find some way to elementarize or decompose overall reactions into a set of elementary reactions, which would be interesting to do and great if we had it, but very time consuming and challenging to do. So as a shortcut, what I thought was, okay, well, I recall that Wolfram and Edda had developed this method to express arbitrary forms of macroscopic kinetic rate laws in the form of this generalized expression. And the advantage of it is that once you have the forward and reverse stoichiometric coefficients, you can express the kinetic rate laws as a form of rapid equilibrium random order mechanism. It works for any stoichiometric coefficient, but here we're just assuming -1 and 1. I'm not 100% sure whether this approach would generalize when it's when the stoichiometric coefficients are not -1 and 1, like 2 and whatever, but I think it probably would. So this convenience kinetic expression can be formulated into a set of exponential cone constraints and use the same optimization approach to hit the boundary. So I was working on the write-up of this approach in January or so this year, and I hit upon a problem. So those of you who are really keen mathematicians might already recognize the problem. So essentially, it's like this. So when we start off and we try to hit the boundary, we're trying to hit some point along the boundary, which is of the exponential cone, this 2D slice. So this red line, or the blue line here. But we can easily find a starting point on the inside if we just take away the objective and we just solve it for a constraint. With just the constraints, we get at some point in the epigraph. But we want to optimize towards the boundary, and that's fine so long as the kinetic constraints are not interfered with by the linear constraints or any other constraints. And so what do I mean interfered with? So one can imagine, and this has happened. I saw this now that happen numerically, is that if once you add on linear constraints in addition to the exponential cone constraints, you could get a situation where your initial vector C, where you're optimizing towards, you might optimize towards the boundary and hit, but you'd hit the boundary of the exponential cone, but you'd hit the boundary of the linear constraints first. And so then that begs the question, well, how does one then find what the correct vector C is, such that you hit the boundary of the exponential cone that's also still inside the linear constraints? And for a number of months, many months, I worked on this, and I'll tell you as far as I've got right now. So I wasn't able to find thus far a single convex function or linear function that would allow me to optimize towards the boundary in one step, like in one conic optimization. So that because let's say in this case here, the line red that's coming down straight towards the bottom, if I just point out like how to avoid optimizing in that direction and always know a priori to optimize in let's say towards the right. I don't know whether such a function doesn't exist. I haven't found it, so I'll tell you how far as I've got. I did find I was able to find a strictly convex function over the epigraph here. So but that function, the way I optimize it is by using a sequence of conic optimization problems. So what I do is I find the initial point, and then I optimize, and then I check or see how far away am I from the boundary, and then I update the linear term. So you can't put this nonlinear conic convex term into the objective, because the objective of conic optimization problem is linear, and I haven't found a way to put that type of thing into the constraints either. You always end up with needing some sort of nonlinear term in the objective, and that doesn't work. So the way I've approached it is by a sequence of conic optimization problems, but because I've found that, let's say this function here, this phi of x is convex over the set, its gradient is strictly monotone. So from any point here, I can rise up along the function or decrease along the function. So I'm not trying to minimize the convex function. I'm trying to maximize it, but I'm going up along the gradient of that, which is anti-monotone. Sorry, excuse me. So because that function on the it lives on a convex set and it is monotone, one can guarantee that you'll always be able to hit the boundary. So while it's not a single conic optimization problem and it's a sequence, it's still really nice because you're guaranteed to be able to find the kinetic steady state provided it exists. So the provided it exists is an entirely other talk, and I've spent quite a bit of time on this. So essentially this exomics to model pipeline produces context-specific metabolic models that admit a thermodynamically feasible solution. And the models that admit a thermodynamically feasible solution are ones that are more likely than not going to admit a kinetically feasible solution. So there is a bit of open work to be done in terms of if you put bounds on the reaction rates and bounds on the concentrations, how will you know for sure that there exists a steady state concentration? That's still an open problem. But we applied this technique to generate dopaminergic neuronal metabolic models that admitted thermodynamically feasible steady states. And I'm just going to show you some numerical examples of running this kinetic algorithm on it. So this is an illustration of a sequence of conic optimization problems. The first red line is the maximum value of the variable, of any variable, and the second red one is the minimum value of any variable, and those are the axes here on the right, the red axis. All the other dots refer to the axis in blue on the left, and so this dopaminergic neuronal metabolic model is 1,200 metabolites, 1,700 odd internal reactions, and 205 external reactions. The total, this 45 conic optimizations solve in about 25 or 30 seconds typically. So that's how long it takes to find the kinetic steady state. The green dot is the merit function that which is the one that tells me how far I am from the boundary. The black dot is kind of like a descent condition, checking to see is each iteration from one to the next descending, and by how much. The light blue dot is showing me how far away the previous optimal solution is from the next one, and the dark blue dot is a measure of how far away from the boundary. The pink one or the magenta one is a measure of the just double checking that I'm indeed dealing with a convex surface. That's kind of a logical check for me to make sure my numerics are correct, because I know it should be mathematically. And the X cross is again a measure of the distance to the boundary in logarithmic scale. So what I see is that I get fast convergence initially, and it keeps going down and down, and then I get quick convergence at the end. So this is pretty typical of linear for first order method. So I think there's possibly capability to increase this speed of this type of iteration by including quadratic terms into each conic optimization step, but I haven't done that yet. So I would say maybe in the end it'll be maybe 5-10 iterations. And the other thing that would speed it up is being able to warm start each conic optimization with the previous optimal solution, which I've seen in previous sequences like that will really speed things up, because it reduces the number of linear solves required for any one convex optimization or conic optimization. So this is just a plot of the residuals. So 10 to the -7 is around where the how far away the numerical solutions are from satisfying a elementary reaction kinetics. And so that's my measure to check to see how far away I am. So for the blue is forward reaction, and orange is the reverse reaction. It's how far away I am from that exponential comb boundary. Interestingly, so in a numerical experiments, what I've been doing is I would set up a situation where I know a solution exists. And so what I do is I create I fix the kinetic parameters. I randomly pick a concentration vector, and then I compute this n times v of f and c, and then I get my dc dt. And then I enter dc dt in with the kinetic parameters also fixed, and I see, can I recover the concentrations with this algorithm? And yeah, I can reliably. And what's interesting from the numerical experiments is that then the net fluxes always come back the same, whereas you can find a kinetic steady state with alternative concentrations that still satisfy the same steady state. So to me, this is kind of interesting, because I hadn't seen this result theoretically before. So a numerical hint, at least for this particular metabolic network, that there should be some results saying that once you've dc dt fixed, that the difference between v forward and v reverse in an elementary kinetics subject elementary kinetics should be identifiable, I guess. Yeah. So this is previous results I had from doing numerical experiments with different sizes of genome scale model. In the time when I hadn't recognized the issue that I needed a sequence of conic optimization problems rather than just a single one. So you can see here, this is a whole body metabolic model, the male version that's been out for some time, and linear optimization for that model takes about 1-2 seconds. Quadratic optimization takes 11 seconds. Maximization of entropy takes 54 seconds. The variational kinetics takes about 200 seconds, and the variational convenience kinetics takes about 239 seconds. So I haven't run the experiments now on when we have a sequence of conic optimization problems, but so you can imagine it could be at worst case maybe 30-40 times those times. But as I mentioned, I think looking towards second order methods and reusing the warm starting each conic optimization will speed it up. yeah, who cares about this? Yeah, no, me, because I like the theory, but ultimately what we need to do is validate that these predictions can predict that this method can predict things that wouldn't been so easy with other methods. And so I'm coordinating a large European consortium where we've now not 25 but additional 20 29 odd different clinical sites collecting blood and urine samples from patients with inherited metabolic diseases, 134 different inherited metabolic diseases. And we lined up 7 or 8 metabolomic labs that are going to combine their platforms to measure first the plasma, and if we find something interesting, the urine of these patients. So about 2,000 patients. So we've started recruiting already, and we're in GDPR, joint controller agreement, administrative quagmire, trying to get all these agreements and everything signed. But yeah, we're making progress on it and hope to have all the ethical applications submitted before Christmas. So to finish on a theoretical note, what I find most interesting about this project is that the series of projects, but really is that we were able to show early on, and I think it's, like I said, I think it's true, that the fundamental equation we're dealing with is not monotone. It's some sort of generalized monotone function, but that's in the ambient dimension. So the lowest dimension you could reduce it to, and it turns out now that when you lift it to a higher dimension, that you can formulate it as like a monotone variation equality, where there are lots of algorithms out there for solving those type of problems, but it's particularly amenable to conic optimization because the exponential cone kind of carries some of the weight for the inner problem, because you have nonlinear but yet conic constraints in each iteration. And I discussed this with some experts in optimization, and I found it interesting for them to say that there's like a kind of a non-result out there, and the non-result, I haven't ever read it in a paper, but it kind of goes like this, and it's to the extent it's kind of like the set of problems that are monotone in the lowest dimension that can be formulated into that are not monotone in the lowest dimension, but can be formulated into a monotone variational equality in a higher dimension by some type of lifting, is not known. Yeah. So it's kind of it's a non-existence result, but the reason why you probably won't see it written down is it depends very much on the structure of the problem, which ones can be lifted into this to make them much easier to solve and which ones cannot. And it seems to me in our case that it's important that there's a duality between the fluxes and concentrations, because that lifting procedure then retains a sort of regularity. So that would be another talk also in itself. But I just put it out there because I think it's super interesting that there are problems that people think are currently insoluble on a large scale that might be soluble on a large scale if they could be lifted to a higher dimension. Yeah. So with that, I'd like to acknowledge my collaborators on this particular project, Michael Saunders, especially, and Inez Thiele for providing the metabolic models for testing, with Thomas Hankemeyer for the support in my career while I was working on this problem, and Professor Paulson for helping me initially get started on this challenge. I also like to thank the ReconFlyMB consortium, who are busy collecting these patient samples, etc. So, yeah, thank you.